Hi, my name is Christina Christensen. The date is July 23rd, 2017. I am doing a persuasive speech on antidepressants encouraging growth of new brain cells for my communications class at Aspen University. Many scenarios were set up as swimming test, chronic mild stress tests for the cause of stress and depression in the brains of the mice and or rats to see if either the drugs clozapine or haloperidol had any effects on their brains. The antidepressants of clozapine and Prozac and antipsychotic haloperidol were tested to see if there was in fact any new growth in brain cells and would it cause the same in humans. I'm not really going to touch on the drug Prozac for the reason being the effects from Prozac were very, very similar to clozapine. So you won't hear much of the drug Prozac within this speech. Clozapine is an antidepressant which has been proven to create new brain cells to help with depression and for the treatment of anhedonia. Anhedonia is not a mental disorder but brought on by depression. Anhedonia is the inability to feel pressure, which clozapine has shown to reverse the effects of this disorder that is caused by depression. Now, when we're talking about anhedonia and the inability to feel pressure, it could be the inability to feel the pressure of food in your mouth, or when to the extreme of not feeling anything during sexual intercourse. Stress and depression treated with clozapine have shown reversed effects of stress and depression. Stress is contributed to the formation of shorter neurons with fewer connections. Chronic mild stress showed behavioral despair, which clozapine reversed. Adult neuroplasticity is comprised of the hippocampal neurogenesis and neuronal remodeling, which is the key role of therapeutic effects of antidepressants. Clozapine was able to reestablish stress-induced impairments in these neuronal structures and gene expressions in the hippocampus and prefrontal cortex. Also, the drug improves neurogenesis, cell survival, and neuronal reorganization. Now, when we get to haloperidol is an antipsychotic which did not have any provable evidence on helping stress and depression. There is a side effect of the haloperidol, which is a sedative effect. So the drug caused a downregulation of adult neurogenesis and neuronal survival, equaling the learned helplessness and aggravation of this drug. Having no flexibility also results in the sedative effect of the haloperidol. The haloperidol had no effect on cell proliferation with no increase in cell survival. It did not, haloperidol had failed to treat stress, which only caused more stress, which led to the aggravation of the depressive-like phenotype because the haloperidol did not help. So, in conclusion, clozapine has proven to be the winning antidepressant with stressed and depressive states and growth of new brain cells. Haloperidol did not present growth of new brain cells and did not solve stress or depression symptoms. Haloperidol proved with not helping just caused aggravation and no need for the fight for survival. I was asked to verbally cite my work that I found through research on the internet. The cited articles I used are antidepressant drug may actually encourage growth of brain cells, retrieved from IFL Science. IFL stands for, excuse my French, I fucking love science. I also used the modulation of adult neuroplasticity is involved in the mood improving actions of atypical antipsychotics in an animal mode of depressions retrieved from nature.com, which is journals and articles. Thank you very much. I hope you enjoyed my speech, and I hope I helped in clarifying how each drug worked and how clozapine came out as the one drug that did help. Goodbye, everybody.